Hello students, welcome back to the channel. In this video, we are going to study detailed structure about alveoli. Now see, you know very well, respiratory zone consists of respiratory bronchioles, alveolar duct and alveoli. So see carefully, respiratory bronchioles is divided into many alveolar duct. And at the end of each alveolar duct, nearly you are having 100 alveolar sac. You can see in this diagram, this is the respiratory bronchiole. Clear, beta? And this is your alveolar duct. This is alveolar duct. So here I have drawn only one alveolar sac, but there can be 100 alveolar sac, and each alveolar sac, this is one alveolar sac, okay, beta? And each alveolar sac will have 20 to 30 alveoli. And they are in how much in diameter? One alveoli diameter is nearly 200 to 300 microns. If you see alveolar sac, diagram is helping you to understand. It gives you a structure of grape-like clusters. Okay, and you will find that the grape-like cluster of alveoli are arranged in the center space. Check this diagram. This center space is known as atrium. Clear, beta? So alveolar sac are grape-like cluster of alveoli which are arranged around the central space called atrium. So atrium is a central space of alveolar sac around which alveoli are present. And you know very well, primary site for gaseous exchange is alveoli. Now, each lungs will contain nearly 300 million alveoli. So if you see, there are so many alveoli and there are so many alveolar sac present, because you know very well, there are nearly 100 alveolar sacs are present at each end of the alveolar duct, which contain nearly 20 to 30 alveoli. So many is present, that is why the lungs have spongy consistency. And this arrangement is just to make for the efficient gaseous exchange, because you know, for diffusion, surface area should be large. So this arrangement is done in such a way that the surface area can be more. And if you see, the surface area of alveoli is nearly 75 meters square. This much surface area they are having. And if you see the diagram carefully, in between the alveoli, you are finding this is pore of Cohen. Okay, you can see these are pores of Cohen, and these are the ones which are present between the adjacent alveoli, which allow airflow between the adjoining alveoli. We say this process is known as collateral ventilation. And this is important to allow the fresh air to enter the alveoli whose terminal conducting pathway is blocked. It might be, suppose, just imagine, suppose this pathway is blocked here. Okay, this path is blocked. It is not working, so it is not able to get fresh air. So this part will get the fresh air from here, that is through pore of Cohen. Clear? So it is helping to allow the fresh air to enter the alveoli whose terminal conducting airways is blocked. Now let us see alveolar walls. What is alveolar wall made up of? If you see carefully in this diagram, you are finding these are your type 1 alveolar cell. Please focus the diagram better. This is your type 1. You can see this is your type 1 alveolar cell. Clear? Which is made up of only flat. Check, this is a flat and this is only nucleus. This is nucleus. And I told you right now the diameter of alveoli is 300 micrometer. And if you see the air which is present here, and this is the blood capillary, the distance between the air and blood capillary is only 0.5 micrometer. Clear, beta? The distance between the air and blood capillary, you can see it is only 0.5 micrometer. So distance is very less between the blood and air. The diffusion, that is why it occurs very easily. So type 1 pneumocytes, they are flat cells, okay, and they are made up of only one layer. The air in the alveoli and the blood in the alveolar capillaries are only less than 5 micrometer apart, so distance has to be traveled very less, and it is an adaptation to increase the rate of gaseous exchange. Then you will see this is a type 2. Check, beta, this is type 2. Check the diagram, this is type 2 alveolar cell. And these type 2 alveolar cells are also called septal cells. They are also called septal cells. And what they're doing it, they are releasing surfactant. They are releasing one surfactant. Now, question come in the mind: ki alveoli is having very tiny diameter. Why they don't collapse under high surface tension? Because there is a thin film of liquid which lines the alveoli, and this film of liquid is always subject to the surface tension. Is it clear? So question is ki alveoli is having small diameter, then also why they don't collapse because a high surface tension is there, because surfactant is secreted by type 2 pneumocyte. Now let us see what is surfactant. See better. Type 2 pneumocytes, actually we have seen also, they are rounded cell, they are not flat cells, making 5%. And they secrete a fluid which coats the inner line surface of these things so that the moisture is there, allow the oxygen to dissolve in that film and finally diffuse to the blood. Reverse for the carbon dioxide. Now they are releasing surfactant also. Surfactant is the one which lowers the surface tension by reducing the tendency of alveoli to collapse at the time of expiration. If you see surfactant structure is similar to the phospholipid. And phospholipid is actually made up of hydrophilic end and hydrophobic end. You know very well. In the cell chapter you have studied, this is the hydrophilic part and this is the hydrophobic part. So they are, see, this is a liquid which is lining the alveolar so that the oxygen dissolve here. So they are making a monolayer. Surfactant is making a monolayer on the surface of the water so that surface tension can be reduced. So they form a monolayer on the surface of the moisture with the hydrophilic head facing the water and tail facing the air. Why they are doing it? Just to reduce the surface tension and prevents the water from causing the sides of alveoli to adhere. Because you know very well, when you do the expiration, this is the one when you do the inspiration, that time your alveoli are fully expanded. But when you do expiration, the alveoli will be collapsing this way. They are touching each other. Okay, so here the water which is present, the thin film of water which is present, we don't want water to stick together. So to preventing the water from sticking together, what we are doing it, we are making one layer that is known as surfactant to reduce the surface tension and prevent alveoli from collapsing when air is exhaled from the lungs. So this much is clear. I hope this topic might be now easy for you. What is a role of surfactant? It is actually reducing the surface tension to prevent the lungs from collapsing during expiration. Now, sometimes what happens, the children which are born early, means nearly the 7th month, or you can say early 8th month, that way, premature babies, we call them, they are generally born with insufficient pulmonary surfactant. And then we can find that they suffer from a disease that is known as infant respiratory distress syndrome. What is the treatment involved? Treatment involved means oxygen is given to the baby and 1 or 2 more doses of the surfactant is also given, which is extracted from the animal lungs. Now, why we are able to understand all this thing? This work was done by one lady in 1950. Okay, it is not there. So she studied his surfactant deficiency can lead to respiratory distress syndrome, a disease which is common in the children which are born in 6 weeks before their due dates. Reason is this key. Actually, surfactant formation starts after 33 weeks of development. And full pregnancy term is 38 weeks. So after 33 weeks, only the surfactant is developed. And if suppose the child is born 6 weeks before, the child is suffering from respiratory distress syndrome. Is it clear? Respiratory distress syndrome. And this work was done by Mary Ellen Avery. And for this, she got the Medical Medal of Science in 1991. Not Nobel Prize, sorry, that is wrong. She received National Medal of Science in 1991. So I think so these two topics are clear, key type 1 pneumocytes and type 2 pneumocytes. Now see, apart from this, they contain dust cell also. These are the dust cell. You can see, check this diagram. This is the dust cell. Alveolar macrophage, also known as dust cell. If you see, in the alveoli, cilia is not there. Cilia is not there. There is no significant air current is there. There is no muco-acceleratory system is working to remove the particles from the surface. So alveoli are actually, they are highly suspected to contamination. Because they lack cilia, they don't have significant air current, so they are highly susceptible to contamination. So WBCs are the one which are preventing the alveoli by engulfing the foreign particles. So alveolar macrophage are the one which are making the alveoli clear from the dust particle which has managed to reach to the alveoli. But if suppose particulate matter which is reaching the alveoli is too much, then defense obviously has to work, macrophages has to work. It can lead to the inflammation and it can lead to irreversible damage. Next important thing is present is of elastin fiber. Next important is elastin fiber. For this, I will come back to the previous diagram. See here. You can check in this diagram, check. This is elastin fiber. Is it clear? This is elastin fiber. Come back to the theory. Beneath the alveolar cell is elastin fiber. And elastin fiber will stretch during inspiration and the recoil during expiration. So this elasticity allows alveoli to expand according to the volume of air which is taken in. You know very well, when you do forceful inspiration, that time your alveoli are fully expanded. So surface area is increased and air is expelled out properly when the elastic fiber are recoiling. So this is going on. But in the emphysema, the problem is elastic fibers are damaged. So in the elastic tissue, they promote elastic recoil when the lungs are stretched. So I hope so in this video, we have tried to cover the topic alveoli. How many are the alveoli present? 300 million per lungs. Okay, they are present at the end of the alveolar duct, making the alveolar sac. Center of the alveolar sac is known as atrium. Lining is done by the two types of cells. One is your type 1 pneumocytes, which are flat cells making 95% responsible for gaseous exchange. Type 2, which is only 5%, they are responsible for secreting surfactant to reduce the surface tension so that the lungs do not collapse at the time of expiration. Because the surfactant is similar to phospholipid like lecithin, it is making a monolayer. Third, we studied that they are having macrophages also known as dust cells, which is responsible for doing phagocytosis if suppose any particle has reached to the alveoli. Because alveoli does not have cilia, neither it has any air current so that they can remove the dust particles. And last is the topic elastic fiber, which is helping the lungs to expand to the maximum when you are doing the forceful inspiration, and because of the recoiling, the air is properly expelled out. So they are responsible for making the alveoli to expand properly and recoil when it is exhalation is going on. And on the outer surface, you will find there is a network of pulmonary capillaries. And you know, pulmonary capillaries is also lined by the single layer of the endothelial cells and the basement membrane present. And you can see it is like a capillary is so thin. Obviously, it is only single layer, so it feels as if the alveoli are surrounded by a sheet of the blood. So I hope so this video has helped you to understand few concepts. Thanks for watching.